This podcast will cover several different types of lipases, and they're classified based on their location first, and then some of their key functions and activity. The first class of lipases are located in the lumen of the small intestine, and they are primarily involved in the digestion of dietary lipids. There are three important lipases involved in digestion. The first one is called pancreatic lipase, which is a triacylglycerol lipase involved in the digestion of triacylglycerol from the diet. It produces monoacylglycerol from triacylglycerol and two fatty acids. The second digestive lipase is Phospholipase A2, and it's specific for the digestion of phospholipids to produce lysophospholipids and a fatty acid. Third type of lipase is cholesterol esterase, which is specific for breaking down cholesterol esters into cholesterol and a fatty acid. Along with bile salts, the digested lipids are integrated into a structure known as a micelle, which allows that micelle to fuse with the intestinal mucosa and allow those latter cells to absorb the digested lipids into the cytoplasm of that intestinal cell. The second type of lipase that is also quite important is extracellular as well, in that it is located or associated with the endothelial wall, and it has access to the lumen of the bloodstream or blood vessels, and can access chylomicrons and very low density lipoproteins in circulation. This lipase is designated lipoprotein lipase, and its function is to break down triacylglycerol found in the lipoprotein particles I just mentioned, and access the fatty acids, allow access to fatty acids released from these triacylglycerols, so the tissues can either use them or store them. Lipoprotein lipase is regulated by insulin. Specifically, insulin increases the expression of this enzyme associated with the endothelial wall. The importance of lipoprotein lipase is that it is the major lipase that allows the tissue to access circulating triacylglycerols from the diet or from the liver, and uptake the fatty acids into the surrounding tissues. Based on their locations, the previous two types of lipases discussed are considered extracellular, one being the first type being present in the lumen of the small intestine, and the second type being associated with the endothelial wall exposed to the lumen of endothelial cells. There are two types of lipases, important lipases, that are intracellular, meaning they are present inside the cell, either associated with the cell plasma membrane or are soluble inside the cells. The first type of lipase is designated hormone-sensitive lipase, and this is predominantly present in adipose tissue. It is a very important lipase in that it can trigger the lipolysis of stored triacyl glycerol, for example, during a fasting state. Hormone sensitivity refers to the fact that this hormone-sensitive lipase is activated by epinephrine binding to the beta-adrenergic receptor on the cell surface of adipocytes. Following a cascade of post-receptor events, this hormone-sensitive lipase is activated due to phosphorylation by a protein kinase called protein kinase A. As a consequence, hormone-sensitive lipase gets activated and will start breaking down triacyl glycerols stored in adipocytes. The 3 fatty acids and glycerol released from adipocytes due to the action of hormone-sensitive lipase are then released into the bloodstream, transported through binding to serum albumin, and will be picked up by various tissues, including the liver that will pick up fatty acids and glycerol. The second type of intracellular lipases are phospholipases, and there are 2 general types of phospholipases that are very important inside the cell. Those are phospholipase C and phospholipase A2. Phospholipases are in general quite specific for unique phospholipids. For example, phospholipase C there is a phospholipase C that is specific to a phosphoinositide called phosphatidylinositol 4,5 bisphosphate, or abbreviated PIP2. The activation of phospholipase C towards PIP2, for example, is dependent on the activation of at least one type of receptor, the alpha-1 adrenergic receptor, that is activated by epinephrine or norepinephrine. The consequence of activating phospholipase C through the alpha-1 adrenergic receptor is to break down PIP2, the substrate, and produce 2 important second messengers initially, diacylglycerol and inositol trisphosphate. Inositol trisphosphate released into the cytoplasm can trigger the release of calcium stored in the endoplasmic reticulum, and therefore calcium inside the cytoplasm also rises as a consequence of activating phospholipase C. The 3 second messengers produced through the activation of phospholipase C will have trigger a cascade of events that produce a variety of cellular responses. The second type of intracellular lipase is designated phospholipase A2. And again, phospholipase A2 is specific for specific phospholipids, a specific head group in phospholipids, among which is the lipid we mentioned before, PIP2. Phospholipase A2 is specific for the 2 position of the phospholipid glycerol backbone. It will hydrolyze the fatty acyl chain associated with the 2 position and release a fatty acid and a lysophospholipid. The activation of phospholipase A2 is a G protein coupled receptor mediated event. That means specific extracellular stimuli will stimulate a unique G protein coupled receptor to activate phospholipase A2. If in the second position of many phospholipids, you will find the fatty acyl chain arachidonic acid. By activating phospholipase A2, arachidonic acid is released into the cytoplasm of cells, and the released arachidonic acid is the precursor for the synthesis of various inflammatory molecules, the eicosanoids, including prostaglandins, thromboxanes, and leukotrienes. The previous 4 lipases discussed were chosen because of their unique locations and unique functions. That is not to say that there are many more additional lipases with unique and important functions in the cell as well. If you like this channel, please subscribe. Thank you.